Welcome to the Deep Dive. Today we are plunging deep into the ancient history of life on Earth, and we're tackling a really thorny problem, figuring out the timeline for bacteria.
Yeah, it's one of the oldest stories on the planet, spanning billions of years. But unlike plants or animals that leave bones or leaves, bacteria are tiny, squishy, and, well, they just don't fossilize well.
Right.
So dating their evolution is a huge challenge.
Exactly. So how do you put a clock on something that leaves almost no physical trace? That's where this fascinating research paper comes in. It takes this challenge head on, blending cutting-edge genomic analysis, machine learning, and geological evidence in a really clever way.
Mm-hmm. Our mission for this deep dive is to really unpack the key insights from this study. We want to understand when and how bacterial life evolved the ability to handle oxygen, that life-changing gas that wasn't always around in abundance.
And how this research sort of reshapes our understanding of bacterial history and, well, the tree of life itself.
Yeah, absolutely.
Get ready, because the methods they used are sophisticated, but they unlock some truly surprising findings about ancient timelines and the deep connection between microbes and Earth's biggest environmental shifts, like the rise of oxygen. Let's jump in.
Okay, so the core difficulty, like you said, is the scant fossil record for bacteria, while things like stromatolites show us evidence of ancient microbial activity.
Like those layered rock formations.
Exactly. They show something was happening, but they don't reveal the specific evolutionary branches and splits within bacteria the way, say, animal bones or plant fossils do.
Right, you see the mat, but not the messy family tree inside it. This is why the Great Oxidation Event, the GOE, is such a pivotal character in this story. Around, 2.315 billion years ago?
Yeah, about then. Earth's atmosphere saw this dramatic, sustained rise in oxygen.
A game changer.
Totally. For a long time, the GOE was seen as the major dividing line, the moment life really had to adapt to oxygen. So, understanding when different bacterial groups evolved, the ability to cope with or use oxygen relative to this event has been crucial, but really difficult.
Okay, so if we can't rely on traditional fossils of bacteria, how did they start building this timeline? They needed a different kind of data, right?
Exactly. They started with a massive genomic data set, over 1,000 bacterial genomes, 1,007 to be exact. And these were chosen to represent a broad swath of bacterial diversity from the GTDB, that's the Genome Taxonomy Database.
Which is already based on evolutionary relationships.
Right. It's a modern framework.
Yeah.
And they use the genes within those genomes to build an evolutionary tree, a species tree,
Okay.
using sequences from 65 well-established marker genes found across many bacterial lineages. These are kind of like the backbone genes that trace the deep relationships.
But genes don't all tick at the same rate, do they?
No, exactly. And evolution can vary across different parts of the genes, so they use sophisticated models to account for those nuances, those compositional variations across branches.
Okay. But even with a solid tree based on genes, how do you put absolute time on it without fossils? This is where they got really clever, you mentioned.
Yeah, this is one of the most innovative parts, I think. They integrated sequences from mitochondria and chloroplasts into their bacterial tree.
The organelles inside our cells.
Yeah. Remember, these organelles inside eukaryotic cells, that's us, plants, fungi, they actually originated from ancient bacteria that got incorporated.
Okay. So they're adding these organelles, which were once free-living bacteria, onto the bacterial family tree. Makes sense?
Exactly. And here's the key. The eukaryotes that contain mitochondria and chloroplasts do have a fossil record, a much better one, in fact.
Right. We find fossils of early algae, early animals.
Precisely. So by accurately placing the mitochondrial and chloroplast lineages within the bacterial tree, they could use the fossil record of eukaryotes to provide calibration points for parts of the bacterial tree.
That's brilliant. It's like using a known milestone in one story to mark a corresponding point in another previously undated story.
Yeah, it's a great analogy. They braced these organelle clades, these groups, to relevant points on the tree corresponding to the fossil evidence for the eukaryotes that acquired them. This provided crucial anchor points where direct bacterial fossils fail us.
Okay, so they've built a gene-based tree and anchored parts of it to time using eukaryofossils. The next step is figuring out what those ancient bacteria were like, especially regarding oxygen. How did they reconstruct the capabilities of ancestors that lived billions of years ago?
Okay, for this, they used phylogenetic reconciliation. There's a tool called ALE they used, and this isn't just like guessing gene presence based on what the descendants have.
It's more sophisticated.
Much more. Reconciliation maps the evolutionary history of individual gene families, how genes were duplicated, how they were transferred horizontally between unrelated bacteria, or how they were lost, all mapped onto that species tree we just talked about.
So it's reconstructing the likely set of genes an ancestor had, even accounting for all that rampant gene-swapping bacteria are famous for.
Exactly. It explicitly handles horizontal gene transfer, which is absolutely fundamental to bacterial evolution and often ignored in simpler analyses. This gives a much richer, more probable estimate of ancestral gene content. It even gives fractional numbers, sort of representing the probability of a gene being there.
And that estimated ancestral gene content is what they fed into the machine learning algorithm.
Yes, precisely. They trained a machine learning model, a powerful tree-based algorithm called XGBoost, which they found very reliable for this task to predict whether a bacterium is aerobic or anaerobic.
What did they train it on? Like, what was the input data for the training?
They used a large data set of modern bacteria where we know their oxygen requirements, pulled data from databases like Bacdive, and included well-known examples like cyanobacteria, definitely aerobic, and typical gut bacteria, which are mostly anaerobic.
Got it. And then they applied this trained model to the estimated ancestral gene sets.
Yep. To predict the lifestyle of those ancient bacteria, those deep nodes on the tree, based on the inferred ancestral gene content, the model predicted their oxygen tolerance.
How confident could they be in those predictions? Predicting billions of years ago sounds tricky.
Well, they validated the model's accuracy on modern bacteria first. For example, they tested it on bacteria from the human gut, which we know is a highly anaerobic environment. It correctly predicted 93% of them as anaerobic. So that gave them confidence it could recognize the overall gene patterns associated with being aerobic or anaerobic.
That's pretty good. I guess my first thought might have been simpler, just look for the obvious oxygen-using enzymes, like terminal oxidases. Wouldn't that work?
You'd think so, but the paper notes that's actually not sufficient. Turns out, some strictly anaerobic bacteria actually have terminal oxidases, maybe for detoxification or leftover from an ancestor.
Oh, okay, so just having the gene isn't the whole story.
Not at all. The ML model's power comes from seeing the whole picture. It learns this complex signature across many genes, not just one or two. It's like looking at someone's whole wardrobe and habits to tell if they're an outdoorsy person, rather than just checking if they own a raincoat.
Because maybe they own one but never use it.
Exactly. The model spots the overall aerobic signature or anaerobic signature in the entire gene content, even subtle things like amino acid preferences and conserved proteins.
Okay, so tree built and calibrated, ancestral gene content reconstructed, and ancestral oxygen use predicted with ML. Now for the big leap, putting the final absolute dates on everything. How did they pin down the billions of years?
That required molecular dating using a Bayesian approach. They used a tool called McMickDate. Bayesian methods are really useful here because they let you naturally incorporate the uncertainty that comes with fossil calibrations and, the molecular clock not being perfectly steady.
Makes sense. What were the key dates they anchored this to? You mentioned the eukaryote fossils.
Yeah, those were key, but they also used others. A hard maximum age for the absolute root of the bacterial tree was set by the moon-forming impact around 4.51 billion years ago. Life couldn't really predate a stable Earth, right? And they also used 15 other calibration points, drawing on established fossil and geochemical evidence at various points across the bacterial tree, things like specific microfossils or isotopic signatures.
And the GOE, the Great Oxidation Event, how did that fit into the dating?
This is where they introduced a really crucial and I think quite insightful calibration. They used a soft maximum age at the GOE, that 2.315 billion year mark, but specifically for nodes on the tree that their ML model predicted to be aerobic.
Okay, a soft maximum. What does that mean in practice? Is it like a suggestion?
Kind of. It's a way of guiding the model without forcing it. It basically tells the model, look, given what we know about Earth's history, it's unlikely that bacteria highly adapted to abundant oxygen are older than the GOE. But, and this is really key, it's soft. Meaning if the genetic data itself and the other calibration points strongly suggest an aerobic lineage is older, the model is allowed to push that age past the 2.315 billion year mark. It lets the data speak, even if it challenges a long-held assumption.
Okay, so they weren't just assuming aerobic life started at the GOE. They were using the GOE as a point of reference to see if the evidence actually supported earlier origins. What was the general impact of adding that soft calibration?
Well, it did what you'd expect for many lineages. It generally shifted the estimated ages of aerobic groups a bit towards the present. Makes sense, right? For instance, the average age of bacterial phyla got about 328 million years younger, when this GOE constraint was applied, compared to running it without.
But did any lineages push past that soft maximum? Did the data say, nope, aerobic life was older in some cases?
Yes, and this is one of their most significant findings. The data did support some aerobic nodes being older than the GOE, most notably the ancestors of photosynthetic cyanobacteria.
The ones that make oxygen.
Those are the ones. Their analysis dated the origin of aerobic cyanobacteria to older than 3.225 billion years ago.
Wait, over 3.2 billion years ago. That's almost a billion years before the main GOE event.
Exactly. It fits really well with growing geological evidence suggesting localized pockets or maybe oases of oxygen existed much earlier, likely produced by those early cyanobacteria themselves in microbial mats or shallow waters.
So the bacteria were making little puffs of oxygen long before the whole atmosphere changed.
Seems like it. And their analysis, based on the presence of key genes related to oxygen metabolism inferred even in that ancient ancestor, robustly predicted the cyanobacteria's stem lineage was aerobic before 3.2 billion years ago.
And they were confident this finding held up. Yeah, it's a pretty big claim.
Yeah, they did their due diligence. They ran checks using different molecular clock models. They varied the specific calibrations for cyanobacteria just to see. The core result, multiple independent transitions to aerobic life occurring before the main GOE remained consistent.
Oh, wow. Okay, so this intricate molecular dating, anchored partly by eukaryote fossils and guided by the GOE, gives us this new timeline. But is there a way to get an independent check on whether this timeline makes biological sense, maybe using those gene histories they reconstructed earlier?
They did exactly that, using a really elegant method that leverages the complexity of gene transfers we talked about.
Yeah.
They used a time-constrained version of that phylogenetic reconciliation model, ALE.
Time-constrained. How does adding time help validate the tree?
Well, this enhanced version of the model incorporates the order of evolutionary splits, the branching pattern, and the dates from their newly dated species tree. And crucially, it applies a fundamental biological rule. You cannot transfer a gene back in time.
Right, that makes sense. Gene transfer has to go forwards or sideways in time, not backwards.
Exactly. A gene transfer must happen from, say, one lineage to another lineage that exists at the same time or later. You can't get a gene from a future bacterium given to one that's already extinct in the past. By adding this time constraint, the model for how gene families evolve becomes much more realistic.
Okay, so the model respects the arrow of time now. How do they use that to test their timeline?
They ran a simulation. They basically created artificial gene family histories evolving on a computer model of a tree where they knew the correct dates.
So they made fake gene data based on a known timeline.
Exactly. Then they took these simulated gene histories and tried to reconcile them to figure out the best history of duplications, transfers, and losses onto different candidate-dated trees. One of those candidates was the correct one the simulation was based on, and others were plausible but slightly different timelines.
Ah, like asking the model, which of these timelines best explains the gene transfer patterns I just simulated?
Precisely. And the result was that the time-constrained reconciliation model consistently gave the highest likelihood, basically the best score, the best fit to the correct dated tree that the gene histories were simulated on.
So the pattern of gene transfers itself contains information about the timing.
Yes, it shows that the messy history of gene family evolution, especially gene transfer, contains inherent information about the relative timing of speciation events. Analyzing it with these time constraints provides a powerful, independent way to evaluate which proposed dated tree is most consistent with the biological reality of how genes actually evolve and move around.
That's a really powerful cross-validation. It's
Yeah.
like the gene histories themselves are confirming that their dated tree aligns well with the complex patterns of gene gain and loss throughout bacterial history.
Indeed. And they also found that analyzing larger gene families, ones with more complex histories, provided a stronger signal for validating the tree. More events means more information.
Okay, pulling all these incredible threads together, the massive genomic data, the sophisticated machine learning predicting ancient lifestyles, the molecular dating anchored by eukaryotes and geology, and this cool validation using gene history, it really paints an epic and surprising picture of bacterial evolution.
It really does. And one interesting confirmation was how well the RID metric, that's relative evolutionary divergence, which the GTDB database uses to classify bacteria based on genetic distance, actually correlates with the absolute geological ages they calculated.
So that GTDB classification is a pretty good proxy for actual age.
Yeah, they found a strong correlation. It means RID is a pretty good stand-in for actual age in bacteria, which is useful.
That makes the GTDB even more valuable for, like, paleomicrobiology. And this whole picture really drives home the difference in timescales between bacterial life and, well, eukaryotic life like us.
Oh, absolutely. What's truly mind-bending is realizing just how ancient bacterial history is compared to ours. The entire domain of eukaryotes, everything from amoebas to animals and plants, is estimated to be around, what, 1.5 to 1.88 billion years old, based on this and other studies.
Okay, that's old, but
But that entire time scale is comparable to the age range of just a single bacterial class in their tree.
a whole domain is only as old as a bacterial class.
Wow. That puts things in perspective.
It really does. And diving deeper, major animal groups like vertebrates or insects, they emerged roughly 650 to 833 million years ago. Land plants, maybe 500 million years ago. Those timescales are similar to the age range of bacterial families in this study, which span from about 450 to 900 million years old.
So many bacterial families we see today were already well established way back in the Archean or Proterozoic eons, billions of years ago.
Exactly. Bacterial evolution wasn't just a slow crawl leading up to eukaryotes. Much of the fundamental bacterial diversity we see today was locked in billions of years ago.
Right. And revisiting that key finding about oxygen. Yes, their analysis strongly supports that aerobic metabolism, the ability to use oxygen, arose before the main great oxidation event. Those early terminal oxidases likely handled just trace amounts of oxygen.
Little whiffs of it.
Probably. And then the GOE created this massive selective pressure that favored the lineages which had already started exploring oxygen use. It drove the evolution of more efficient systems.
So they weren't just passively waiting for oxygen to appear. They were already interacting with trace amounts, maybe in those microbial mats, and the GOE was like hitting the gas pedal on aerobic evolution for those groups.
That's a good way to put it.
Did they see how often lineages switch between using oxygen and not? Was it a one-way street?
No, not at all. The model inferred a lot of flexibility. It estimated something like 84 transitions from anaerobic to aerobic lifestyles, but also 66 transitions going the other way, from aerobic back to anaerobic throughout bacterial history.
So losing the ability was common too.
Apparently, these transitions weren't just clustered around the GOE. They happened repeatedly, likely as environmental conditions changed, new niches opened up, or old ones disappeared. And again, that ancestor of cyanobacteria and its close relatives was robustly predicted to be aerobic way back before 3.2 billion years ago.
That flexibility makes a lot of sense for adapting to a changing planet over billions of years. Did the study shed light on the pace of these changes or the overall pattern of diversity?
It did, yeah. They looked at rates of gene content evolution, how quickly genomes change by gaining or losing genes. They found that rates of gene transfer and gene loss are significantly higher, like orders of magnitude higher than rates of gene duplication in bacteria.
So borrowing and discarding genes is a much faster engine of genomic change than just making extra copies of your own genes.
Absolutely. It really highlights the importance of horizontal gene transfer. And looking at the overall pattern of gene content diversity over time, how different bacterial genomes are from each other, they found strong evidence for what they call an early burst.
An early burst, meaning most of the variety appeared early on.
Kind of, yeah. Most of the variation in gene content we see across all bacteria today seems to have been established very early, before 3 billion years ago.
So a massive explosion of genetic innovation way back then.
Seems like it. It fits with this idea of an Archean genetic expansion. This initial explosion of diversity was associated with the radiation of major groups like Terrabacteria and Gracilicutes, basically exploring a wide range of metabolic possibilities very early in life's history.
And did they see periods later on where certain lifestyles had an advantage?
Yes, they identified periods where aerobic lineages seemed to diversify faster, which, not surprisingly, correlated pretty well with known increases in atmospheric oxygen, like after the GOE and later pulses.
That seems intuitive. More oxygen, more opportunity for oxygen-using life to spread and diversify.
Right. But interestingly, they also found evidence for a surprising late reversion around 200 million years ago. It looked like anaerobic lineages actually showed an advantage in diversification rate.
200 million years ago. That's almost yesterday, in geological terms, way after the big oxygen events. What could cause that shift back towards anaerobic life gaining an edge?
Well, the paper suggests it might be linked to the emergence of new, widespread anoxic environments around that time. Think about the diversification of early animals creating complex gut tracts, perfect anaerobic niches.
Like inside dinosaurs or early mammals?
Could be. Or maybe new environments associated with decaying plant matter as forests became more widespread on land. These created new, rich anaerobic niches that favored the evolution and diversification of those lineages capable of exploiting them.
That's a fascinating connection. Bacterial evolution being shaped not just by geology, but by the evolution of other life forms and the new environments they create, like feedback loops.
Exactly. And finally, the study offered some insights into specific molecular adaptations beyond just the core energy pathways. They found evidence that enzymes involved in managing oxidative stress, dealing with the toxic byproducts of oxygen.
Like antioxidants.
Sort of, yeah, things like catalysis and superoxide dismutases, and even components of the fundamental protein translation machinery, the ribosomes, show signs of having adapted as oxygen levels changed throughout Earth's history.
So the rise of oxygen didn't just mean developing new ways to get energy. It meant really fundamental cellular machinery had to evolve to cope with this reactive, potentially toxic gas permeating the environment.
It really underscores just how profound and pervasive the impact of oxygen was on life, right down to the most basic molecular level. It touched everything.
Wow. This deep dive, leveraging these incredibly sophisticated methods, combining genomics, machine learning, geological context, has painted a truly rich and surprising picture of bacterial history. It really feels like it shifts our understanding of key events like the origin of aerobic life and when the major branches of bacterial diversity were actually laid down.
It's a powerful testament, I think, to how we can reconstruct the deep past, even for life forms that leave minimal traditional fossils, by integrating different kinds of data and methods in these really creative ways.
So what does this all mean for you, listening? If these ancient microbial adaptations, forged billions of years ago by global changes like the rise of oxygen, truly laid the metabolic foundation for almost all life that came after, including our own, well, it makes you wonder, what other fundamental secrets about the earliest chapters of life are still hidden, waiting to be uncovered within the genomes of the microbes living all around us, and even inside us today?